Hi. Thank you very much for attending this webinar. I'm Paul Avillach from Harvard Medical School, and there's also Jamie Esposito, Espino from the Causal Discovery Network from Pittsburgh, University of Pittsburgh.
Hello.
So the purpose of what we want to show you today is the results of ongoing collaboration between our two BD2K Center of Excellence, the CCD Center for Causal Discovery, and the picture from HMS, in order to have API working together to make biomedical discoveries. So the overall context of this project is where we have a picture API that enables to have access to any kind of patient-level data. We expose it to using our API, and we expose it so that the center is using an authenticated and authorized method in our API, so that Jeremy and his team from the CCD project can have access to our data, run the CCD analytic framework to infer new discoveries, and that we show back as a visualization within our network. So the overall goal is to have our two projects working together, only connecting using API and not sending the raw data as we usually do. So the presentation will be in two phases. The first one is I'll show you what comes out from the picture API from a very specific data set, autism, the Simon Simplex autism data set. And then afterwards, Jeremy will show you from the causal graph analytic framework that goes on in Pittsburgh, and then how using our data set that was exposed through our API, he was able to create first inference based on the data. So I'll get started on presenting you the picture framework, where what we created is on having on Amazon an HIPAA-compliant infrastructure, where we have the I2B2 model in order to store the data, the transport application to have a user interface to have access to the data, but also creating a RESTful API to also access this full data set using authenticated and authorized method. Then we're using the Jupyter notebooks in order to deploy the infrastructure and to deploy the source code of how to run an analysis to be able to do reproducible science, and using the Docker component to make sure that we could definitely create something scalable on Amazon. So the overall goal of our API is to have any kind of patient-level data that can be clinical data from EHR, from registry, or exposition, exomes, a full genome, that can be accessible to be able to have access to this data set to make correlation. So you can play with the API from this link, bd2k-picture.hms.harvard.edu, and the source code for the API is here. And the demo I'll be showing you will be using clinical data that is from one database with exome annotated from another one, using also the exact exome aggregation consortium database that we made also available through the API, and all this running on our Jupyter notebook. And this connection enables to have access. And because the data set we'll be using is protected by an IAB, it's patient-level data on an autism cohort, we needed to enable an authentication and authorization mechanism using our API. And so we use O0 as a federated identity provider management tool that enable us to have many different identity providers, so that we don't store any login or password. What we store is only the fact if a login is authorized to have access to a component. And so that's why we enable using the ERA Commons system with all the NIH email in Commons Federation, HMS, BCH, and of course Pittsburgh, and also public identity providers like Google, Facebook, so that you can first be authenticated and then authorized, meaning that you have access to some kind of data. And what we also, and this is enabled through our user interface of I2B2 Smart, but also using our RESTful API. And you'll see during the live demo how this works. And also using the Jupyter Hub network, we wanted to be able to have for different investigators to deploy Jupyter containers directly. So this is using from one EC2 on one virtual machine on Amazon, where we have a Jupyter Hub container that deploys every time there's a new user. You have a new container which is created that enables to run those Jupyter notebooks. So those private research environments are completely separated from another user. So if they break it, it won't break the overall infrastructure. So that's the whole idea of scalability and reproducibility using the Jupyter notebook. So now looking at the data that we've been using for this use case is the Simon Simplex collection autism data, where we have 2,800 simplex families, where we have one proband that has autism, and we have data on at least one unaffected sibling, but there can be many other ones, but there has to be at least one, and to have the both parents. So we have the full clinical data, more than 6,000 clinical variables for each proband. We also have clinical variables on the family, and most important, we have all the SNP arrays and full exomes on the full quad to have a very rich data set with all the clinical and genomic data available on those patients. So the clinical data is divided in 27 standardized questionnaires. For example, the first one, ADIR, Autism Diagnosis Interview Revised. This is a 90-minute questionnaire. There's 90 questions, and there's 27 of the standardized questionnaire that have been integrated and available through our API. So the driving use case I'll be showing you is something that was discovered using this data set, and a finding that was already published, was the fact that when you have a CHD8 gene mutation for this proband, you have a larger head circumference. So you have here the head circumference with Z-score and a larger head circumference for all the probands when you have a CHD8 gene mutation. So it's an example of going from a genotype to the phenotypes of making where it's known to have a causal relation between a gene and a phenotype. And with this driving use case, I'll show you now a live demo using both tools. The first one is the user interface of I2B2 Transmart that we developed for the Picture BD2K project, where you have this data set, and I can, as I showed you, we're using Auth0 to have this management of all the identity providers. So I'll be using my, for example, HMS ID. And by logging into this platform with single sign-on, I now have access to all this data for this project. And I have access to all the clinical data, the 2,600 questions, but also the different mutations. So for example, CHD8. So I can compare between patients that are with 8 patients known to have at least one mutation in the CHD8 gene versus 2,500 patients not known not to have a de novo mutation in CHD8. So what I'm creating is two cohorts, and comparing them. On the left, I have CHD8 gene mutation and CHD8 gene mutation, and on the right, with no CHD8 gene mutation. And so you can see the comparison of age between both groups, sex, and race. Now, with those two cohorts, I want to take my clinical variable and I have access to the 6,000 clinical variables, drilling down to the most commonly used variables. For example, the head circumference. I can drag and drop this variable, and automatically I will have the breakdown of this variable the histogram of the head circumference for all the probands that are part, so all of them, and now how they're divided into those two groups. On the left, when you have CHD8, yes, here, and on the right, CHD8, no. And you have the average of the head circumference, so 56 versus 53.98 centimeters between both groups. And is this difference statistically significant? Yes, with a t-test at 3, a p-value at 0.018, the result is significant at the 95% confidence interval. So this user interface is to generate hypothesis. It's to have a very quick sense of touching the data. Is this completely irrelevant? Of course not, because I didn't do a breakdown stratification per age, because the head circumference of all kids put together doesn't mean much, but this is just a very rough estimate how we can compare between showing a correlation, a causality correlation between CHD8 gene mutation and the phenotype, the head circumference. So the thing I'll ask you to remember is this p-value, 0.018, and now I'm going to show you how using a RESTful API, I can redo the exact same analysis on the exact same data set. So using our API, I'll first start using a tool called Postman that enable us to launch a arrangement and now it should be bigger. Where using this tool Postman, I can run the API queries. And before that, I need to go from the user interface. And because I'm authorized as a level 2, I have an API token, what we call a BT2K picture API key. This key is valid for 24 hours to make sure that I'm authorized, and this key will be set in my user interface to be able to, here, when I'll be creating an API call, to have access to the data. And now the first step is to start a session. And now I'm authorized, I'm authenticated and authorized to run queries. Based on this, I can look at what all the resources are available from this API installed on the Simon Simplex project. So there's two resources available. The first one is Exact, which you can run all the different queries looking at the chromosome position, the variant, and to be able to have the minor allele frequency. And the second resource available is the I2B2 Transmart data from Simon Simplex, which is the exact same database I showed you and run from the user interface. So it's the same Oracle database that is available. And those are all the different elements I can use in my query to have access to the data. So constrained by value with dates, strings, operators of equals, bigger, like, the different unit of measures, the different time that are integrated, the different date format. All this is available through the API. And now I'll be running a query where the first query will get, now I'm doing a post, and from this post, I'll be selecting, asking to get all the patients where I have the head circumference, and also with extracting the CHD8 status. So by using and pushing this JSON script, a file that will be sent using the run query, and now I just run it, and I have a result ID. So this is the ID of my data of the query that was generated. So 96,701. I do a copy, and I want to look at the result status from this query, because some query can take some time. So that's why we have an API to make sure that the query is, the results are available. And now it's telling me that those results are available because it was a simple query, so it was very quick to get a sense out of it. So now what is the data, the available format to extract this data set? So by adding here all the available formats from this data set, I can extract it in JSON, XML, Excel, and CSV format. And now extracting this data set for example, in CSV format, run query, and I'll have here always there's the patient IDs. And because I ask to have access to the CHT8 status and the head circumference, so in here I have the head circumference of those patients. So if instead of having this, I want to have the result not in CSV but in JSON format, I have now all the results in a JSON format. So that's the API working, doing a step-by-step of all the different elements. Now, by doing again a copy and paste of this API key, using the Jupyter notebook as I showed you, we have users on this Jupyter notebook per investigator. So we have admin. I can see there's one account, so it's one separate Docker container per user to have access to their private research environment. So I'll access mine, Avillach. So this is running in one Docker, and looking at this project, now I'm using a Jupyter notebook that will run the query to show you all the different steps using R to have access to this clinical data and to reproduce the comparison between CHT8 and the head circumference. So the most important part is when you run the API using the keys to have where you are, what data set you are accessing, for example here, Simon Simplex dataset, and then head circumference using this API token, and then what I'll be doing is to extract the CHT8 status for all those patients and extracting the head circumference, the CHT8 status, and then running a query, the different available format where I can download the data into a R frame, and then the most important here, running a t-test where I have the t-test of the head circumference by the CHT8 status, and we get, with no surprise, the same result with the same p-value of 0.018 as I was getting from the user interface, because here I'm accessing the exact same data set which is stored in Oracle. So this is a very simple example of how we can access all the clinical data and our omics data from the Simon Simplex dataset using our API. So this was the first step in the context of this and making all this available for Jeremy and his team to be able to access our data and into, so what I just showed you was here, this picture API, and how the CCD team in Pittsburgh used our API and their hard path, of course, to develop it and to extend it so that it can be really useful based on their use case, and exposing the Simon Simplex dataset that I showed you how using the I2B2 Transmart user interface we were able to do a query, but also from the API. And now I'll give the floor to Jeremy, where he'll be presenting you on his side exactly what happened in order to reuse all this data that we made available through our API. So now, if you can make Jeremy as a presenter. Hello?
Okay. Yep, I'm going to share my screen. It's showing up?
Yep.
All right. Okay. Okay, thanks, Paul. So all the work that Paul and his team performed to make this data accessible is something that we want to use to do causal discovery. And as Paul had said in his discussions, the goal of our collaborative work was first to create the necessary APIs and deploy those APIs and infrastructure on available resources. So we began our deployment and development on Pittsburgh supercomputing resources here in Pittsburgh, where we have available, for example, 3 terabyte 64-core machines and even larger machines, 12 terabyte machines with over 80 cores. So with that type of horsepower, we can do some very nice computing. As a goal for the supplement and our collaborative work, we wanted to show how we could deploy our causal discovery algorithms on a cloud-based resource. So what you're seeing here is a web application that's been deployed at Amazon. And what we have is a web server which hosts our application. And then that web server is connected to an on-demand high-performance computing cluster at Amazon using the resources at Amazon. So we can use the resources on the cloud for those available large memory machines. We also have the ability to switch to our resources over at Pittsburgh Supercomputing Center for even larger machines, as I said, 3 terabyte, 12 terabyte machines. So let me give you a brief demonstration of what we're able to do with this application and perform some causal discovery. So our application comprises data management functions, access to causal discovery algorithms, managing job queues, and then viewing results. Make this a little bigger. So here I've imported several data files that we're going to play with. I'll show you a small one first. This particular data set called retention deals with college acceptance and college graduation that was published by a colleague of mine, Marek Drutzel, in the early 2000s. But what I can do here is I can take an algorithm, one algorithm called fast greedy search, and take the data from this particular U.S. news report on college acceptance rates, and then our causal algorithm. What are the relationships, causal relationships, between variables in the algorithm? So I run this over at Amazon. It's running right now. And then I can take a look at the results from that causal analysis. So what we're showing here are causal relationships between the variables in that data set. The first thing we see is spending per student influences rejection rate. So directed arcs show that there is some causal relationship between these variables. The algorithms in our system also allow you to impose constraints on the search. So I can take the same data set and impose some temporal constraints. So I'll do that here. And again, we get a causal graph. And something that might be interesting to educators is if you impose some temporal constraints to this, that faculty salary is related to student acceptance rate and better test scores, and also spending per student influences rejection rate, and also test scores influence graduation rate and class standing. So all these make sense. We can take larger data sets. So one of the data sets that I've imported into our system is a small breast cancer data set that's abstracted from the Cancer Genome Atlas. And I'll run an algorithm on that, again, fast greedy search. Hold on a second. I'll impose a constraint that only genes can influence the expression of RNA using this prior knowledge file. Set the default and run this. And if you're familiar with cancer or breast cancer, you see that our algorithm has pointed out that, let's see if we can move back here, that the cancer driver TP53 has many influences on the expression of of RNA in our data set, which is completely consistent with what we know about the biology. So what we can do, because we created APIs, is use the API that Paul's team has created to extract data. For example, the Simon simplex collection data that he showed you just prior, and use our causal discovery algorithms to generate new hypotheses about our data sets. So I'll show you a complete demonstration that's run completely on the cloud with regard to APIs, and then show you a causal graph from the data that I extract. So similar to what Paul just did, I'll log into the picture instance, and I'll grab an API key. And then I'll use that API key to paste into my Jupyter notebook. This is a Python example. So here I place my API key into my script. And what I'm going to do here is make a request for data from the Simon simplex collection picture instance that Paul demonstrated. And then I'll take that data and then feed it via our API into a causal discovery algorithm. So here I'm using my API key. And then what I'll do next is create a query similar to the way that Paul created a query in R, and also showing you in the Postman application. So I build my query, and I post my query to his system. I check to see the status of that query. It says that it's available. And I grab the results. And the results are formatted as a comma delimited file with a number of instances. For convenience, I'll save this file so that I can scrub it and take out missing values. And now I'll take that data and process it through our causal discovery algorithms. So in our API, we make use of obtaining an API key via SSL encrypted basic authorization. I'll do that next. Now I have a web token with which I can perform additional operations. So I upload the data that I obtained from the picture instance to our system. Now I've just uploaded it. I tell our system to review the data in that file and label it. And then I get to the point where I call on an algorithm. So here I'll be using the fast greedy search algorithm and have it use that particular data file that I provided. Again, this works from the perspective of a high-performance computing job. Over at Amazon, what's happening once I tell the system to execute the analysis is that the on-demand high-performance computer will allocate a node and then run the algorithm on that node. I wait until the job is complete. Oh, looks like it's going. Let me see what happens here. may have finished. Let me see. Okay, for that particular job, okay, so this just ran. So the results of the analysis for the particular variables that I've selected from the picture ID are showed as directed arrows. For example, age influences comp score, age influences VMA, nonverbal IQ influences comp score, nonverbal IQ influences patient hum. And what I can do is I can go back to my web interface and take a look at the algorithm results. So this is the job that just ran, and then look at this from a graph perspective. So what we plan on doing as a result of the availability of the Simon Simplex collection data and autistic and neurotypical patients in the Simon Simplex collection from pictures, it makes it very easy for us to extract data and query for data in this particularly large data set and perform causal analysis on this data. And we'll be looking at replicating, for example, what Paul has found in the head circumference CHD8 example. One of the things that I've been asked is what do you feel is the most important in your collaboration that allowed you to use these APIs and work together? And I'll have to say that to the hard work of both our teams, what made the most difference is the availability of well-documented APIs. So here's the API for picture and the API for our causal discovery algorithms over the web. And not only making an API for our teams to use, and its documentation was most important, but also working examples. So when we put together not only our documentation, not only the working APIs that are deployed to Amazon, but also the examples that are embodied in a Jupyter notebook. And the Jupyter notebook is really easy just because it's very shareable, very demonstrable. We can really work together well. And so the takeaway lesson was when we were creating these APIs, document it well, and then also provide working examples that you can integrate into your own work. Okay, so that's my demonstration, and if there are any questions for Paul and I, we can entertain them.